cell division. There are two kinds of cell division, as you know. One, mitotic cell division. And two, meiotic cell division. Cycle and cancer. Okay, in eukaryotic cells, there was an organelle which is called nucleus. And that nucleus, that organelle, which is called nucleus, contains the structures called chromosomes. And those chromosomes contain the DNA, which is genetic material. All right. Now I'm going to start with explaining you the connection between DNA, chromosome, and the cell. So let's genetic material in the cell. These are organisms. So these are two people, and every organism, every multicellular organism is composed of cells. We call them somatic cells. Inside the cell, there are organelles, and one of these organelles is nucleus. In the nucleus, there are chromosomes. For example, in a human cell's nucleus, there are 46 pieces of chromosomes. Keep in mind, in addition to nuclear DNA, in addition to DNA in the chromosomes, which are located in the nucleus, there are DNA molecules in the mitochondria as well. But we are not going to concern about that today. Today, our subject is especially focused on chromosomes in the nucleus. All right, what there is in the chromosomes? First of all, these chromosomes are seen as chromatins, structures called chromatin in the normal cell, in a normal non-dividing cell. But what is there in the chromatin or chromosome? Let's look at the molecular structure. The chromosomes consist of DNA and protein, two kinds of molecules, DNA and proteins. But there are a lot of different kinds of proteins, especially histone proteins. The special types of proteins, which are called histones, are very important for chromosomal structure of chromosomes. Now, when we look at the structure of chromosome, and when we separate those molecules, we have DNA. Every chromosome is another DNA molecule. So one chromosome means one DNA molecule. That means we have 46 DNA molecules in our cell's nucleus. In a human cell's nucleus, we have 46 molecules of DNA, 46 pieces of DNA. And this DNA is packed with the proteins, which are called histones. So this is our DNA. So this blue thing is the DNA. And we take histone proteins, we put them together, and we wrap the DNA around those histone proteins like this. So this is our DNA. This is the DNA. And we pack the DNA around the histone proteins, and we get a beads on a string model, beads on a string structure here. So these are the beads, and think like this is the string. And then we pack this structure on itself, and we get more condensed, we get a more condensed structure. So if you look at the diameters, DNA's diameter is 2 nanometers. This beads on a string structure's diameter is 10 nanometer, and the solenoid structure, which is more condensed form, is 30 nanometers. So this is the chromatin in a non-dividing cell's nucleus. So if the cell is not going to divide, DNA is found as chromatin structure, which is this. And every single DNA molecule is another piece in the nucleus like this. Now, when we look at the chromosomes, one chromosome is one DNA molecule, I said. One chromosome is one DNA molecule. And there are special regions on that DNA molecule, on every single DNA molecule. For example, now we are looking at the chromosome 11, human chromosome 11. This chromosome is a packed DNA molecule. If we open it up, if we clean the proteins, we have a long DNA molecule. And on this DNA molecule, there are special regions which are called genes. So this part of the DNA is one gene, this part is another gene, this part is another gene, this part is another gene. Genes are the DNA regions which are responsible for synthesis of a protein or an RNA molecule. Okay? Genes are the DNA regions which are responsible for synthesis of a protein or an RNA molecule. So genes are the DNA regions which encodes the proteins or RNA molecules. As you can see here, on the chromosome, on one DNA molecule, genes are short regions. But the DNA is continuous. Between the genes, there are the regions of DNA which are not genes, which do not encode any proteins or RNA molecules. These are just DNA. They are not converted to proteins. They are not responsible for any proteins. Also, there is another description we have to make for the genes. One gene is also responsible for a genetic character. So our genetic characters, our phenotype, is determined by the genes. Some of the genetic characters are determined by only one gene, but some of our genetic characters are determined by 2 or more genes. So what are those genetic characters? Those are like your blood group. For example, ABO blood group is one genetic character. Our age group is another genetic character, or color of your skin is another genetic character. If someone has a genetic inherited disorder, for example, thalassemia or anemia or SMA, that's another genetic character. So to have a inherited disease like thalassemia, to be a thalassemia patient, to be carrier of thalassemia is one type of that character, but to be normal for that disease, not to have that disease, is another form of that character. So these are the genes responsible for our genetic characters. For example, on the 11th chromosome, on this region, look at these genes. These are globin genes. This is beta globin. This is delta globin, alpha, A gamma globin, and epsilon. So this beta globin this beta globin gene is responsible for production of hemoglobin's beta chain. So hemoglobin protein contains 4 peptide chains, and 2 of them are beta peptides, 2 of them are alpha peptides. So this beta gene is responsible for production of that beta gene. So every human knows how to synthesize that beta peptide, that beta globin peptide. How do we know that? We know that because we have the DNA encoding that peptide. So on the chromosome, on the DNA, there are special regions called genes, but there are DNA regions between these genes. How do we call them? Those are called intergenic regions, okay? Genes and intergenic regions. Genes are responsible for our genetic characters or synthesis of a peptide or synthesis of an RNA molecule, but intergenic regions are not responsible for synthesis of any RNA or protein or any known genetic character. So on the chromosome, on one DNA molecule, there are regions called genes, and between the genes, there are intergenic regions, but DNA is continuous. All right, now let's go inside. So this is another diagram showing the chromosome 11. So this is chromosome 11. Now we are looking at only this part, or let's look at this. This is the chromosome 11, and this is the zoom scale. Look at the zoom scale. If we zoom out, we can see almost the whole chromosome. And on this chromosome, there are a lot of genes. There are a lot of genes on 11th chromosome. If we get closer, now we are looking, so look at the zoom, scale. Now we are more zoomed in. So there are, now we can see the genes separately. So hemoglobin beta, hemoglobin delta, pseudo beta, G gamma, A gamma, and epsilon. So these genes are ordered on chromosome 11's this region. This is only, now we are looking at only this region of chromosome 11. And the order of these genes is constant in every human. Every human have these genes on chromosome 11 in the same order. All of us have the same genes in the same order and only on chromosome 11. Is there any difference? There are small little differences, but if you are a human, if an organism is a human, that organism must carry these genes on chromosome 11's this position, P15.4 position, and in this order. Now let's look at that closer. Now we are closer. Look at the zoom scale. So we were here, the whole chromosome 11. Now we zoomed in. Now we see 1, 2, 3, 4, 5, 6, 7 genes. And this group of the genes is called beta globin gene family. Now we are, so look at the scale, zoom scale. Now we will zoom in more. Okay, now we zoomed in more. Now we are looking at beta globin gene. Now we see only beta globin gene. So this is our beta globin gene. Where is it? Its position on the chromosome 11 is stable in everybody. Every human must carry this specific gene at the same position of chromosome 11. That's the same for all other genes. We have 20,000 different genes in our genome, and all of them must be at the same position of the same chromosome in everybody, every human. All right, this is beta globin gene. Now, so there are other genes as well. So this is beta globin gene, which is 2 kilobases, around 2,000 base pairs. So beta globin gene is a piece of DNA which is 2,000 base pairs. So ATG, CTG, TTA, like that, 2,000 bases. There are genes bigger than this. For example, BRCA1. It's about 80 kilobases. 80 kilobase means 80,000 base pairs. MYH7 gene, which is 20 kilobases. So these are different genes in our genome. So if we look at in more detail, now we are closer to the beta globin gene. Let me see if there's any other. If we look at the beta globin gene, more details, we just see the nucleotides, the bases, DNA bases, if we look more closer. And these are the DNA sequence. This is the DNA sequence of beta globin gene, human beta globin gene. This is almost the same in everybody, but there are small little differences between those single letters. There may be small little changes between these base pairs when we compare the people, and they may cause a phenotypic change, or they may not. They may not affect the phenotypic change. If they affect the phenotype, and if they cause a disease or death, then we call it a pathogenic mutation. Those pathogenic mutations cause diseases. But if it doesn't cause any disease or death, then we call it polymorphism or variation. All right. So these genes on the chromosome, every gene is responsible for production of another peptide. And these peptides may work by themselves as a single peptide protein, or they may get together and they can make the quaternary structure of proteins. And these proteins may be responsible for metabolism of the cell signaling, motility, mitosis, cell adhesion, or stress response. When we collect these cell functions, so proteins determine the cell functions. Genes encode the proteins, proteins determine cell functions, and cell functions determine the organism's phenotypes, organism's anatomy, physiology, and behavior. And collection of all of these is the organism. So, okay, did you make the connection? If you don't understand the chromosomes, you will never be able to understand mitosis or meiosis. Now, there are some terms I want you to learn right now. Chromosome, homologous chromosomes, chromatid, sister chromatid. And chromatin. Now, I already showed you the connection between chromatin and chromosome. Now, the cells are found in different stages of their life cycles. If the cell is not dividing, that means that cell in interphase. So the cell doesn't have to divide. But if the cell exists, that means that cell is produced by division of a previous cell. Now, in a non-dividing cell, DNA is found in the nucleus as chromatin, the thin structures. Let's make another work page. So, let's say this is our chromatin, okay? This is the chromatin. But from each chromatin, so each chromatin means a single chromosome. In non-dividing stage, if the cell is not dividing, the chromosomes are found as chromatins. But when the cell is going to divide, the chromatin becomes chromosome, which is a thicker and shorter structure like this. And the chromatin becomes a chromosome. So this is chromatin, this is chromosome. So that means every chromatin is one chromosome. Only difference is chromatin is thin, chromosome is thick. So when we say chromosome 1 during interphase, that's in chromatin structure. So now, in the human cells, there are 46 chromosomes. In every single somatic human cell, there are 46 chromosomes. That means we have, if it's not dividing, we have 46 pieces of DNA found in chromatin structure. Now, let's say this is a diploid organism. But in diploid organisms like human, the chromosomes are found as pairs. So there are 2 copies of every chromosome. So this is another chromosome, and this is another chromosome pair. Okay? So the constant thing is, on these chromosomes, always positions of the genes are constant. For example, there is gene 1 here, there is gene 1 here too. If there's gene 2 here, gene 2 is here, gene 3 here, gene 3 is here. So everybody has, or every cell of a diploid organism has 2 of each chromosome. So this is gene 1, this is gene 2, and this is gene 3. Then the same genes are exist on this copy of the same chromosome too. 1, 2, and 3. So these are the genes. So, and we call this chromosome 1. This is our chromosome 1. It's too difficult to write with mouse. And this is chromosome 2. And this is chromosome 3. All right. So gene 1, 2, and 3 are on chromosome 1. Gene 4 is located on chromosome 2 for, let's say, 5 and 6. Of course, 5 and 6 and 7, 8, 9, 7, 8, 9. So gene 7, 8 is too difficult to make, and 9 are located on chromosome 3, 7, 8, and 9. Okay? Did you understand? Does it make any sense? So we have 2 copies of each chromosome, and the same thing with these chromosomes are order of the genes. Every chromosome carries the same genes at the same position. So if this is human chromosome 1, every human must carry gene 1 at this position, gene 2 at this position, and gene 3 at this position. So every human carries these genes on chromosome 1 at these positions. So since one human carries 2 chromosome 1s, 2 of the chromosome 1s, it has two copies, that human has two copies of each of these genes. Now, these genes may have different versions. We call them allele. For example, let's say this gene is responsible for blood group, ABO blood group. So blood group can be A or B or 0. This person may have A gene here, A type of blood group gene here, B type of blood group here. Or both of these chromosomes may carry the A type of blood group gene. And in here, the other gene may be responsible for the eye color. This gene, which is responsible for eye color, can cause blue eye or brown eye. So one version of this gene causes blue eye. The other version of the eye color gene causes brown eye. But those are still the eye color genes. Those are still the gene for eye, responsible for eye color. But there may be different versions. So there are chromosome 1, then there are different chromosome 1s of different human chromosome 1s. But they must carry the same genes, but they may be in different combinations. For example, A blood group, blue eye. Okay? Another chromosome 1 may carry B blood group and blue eye, or another chromosome 1 may carry B blood group and brown eye. So these makes different versions of human chromosome 1. Okay? Did you understand? Am I making sense? Buraya kadar herkes anladı mı? Bir de Türkçe anlatmamı isteyen var mı buraya kadar? All right, let's go ahead then. Now, this is a non-dividing cell and these are the genes on chromosomes. Now, let's divide this cell. So, this is the chromosome 1 pair. This is the pair of chromosome 1. This is the pair of chromosome 2. And this is the pair of chromosome 3. And do you know why do we have 2 of each chromosome? Why do we have 2 chromosome 1, 2 chromosome 2, 2 chromosome 3s? We have 2 of each chromosome because we are diploid organisms. In diploid organisms, one of each chromosome comes from the mother, is originated from the mother. The other one is originated from the father. Hocam ama bunlar otozomal değiller mi? Evet, bunlar otozomal otozomlar. Şimdilik onları gösteriyoruz. XY kromozomları şimdilik geçmeyeceğiz. Biraz sonra geçeceğiz. All right. These are, one of this is originated from the mother. The other one is originated from the father. Here, the chromosome which we take from our father is called paternal chromosome. The chromosome we receive from our mother is called maternal chromosome. 2 new terms for you. So one copy of this chromosome, one copy of each chromosome pair is paternal. So we show paternal here. And the other one is maternal. Paternal and maternal. Paternal copy and, oh my God, maternal copy. Okay. So now, at the moment, the cell is interphase. And this cell decided to divide. This cell has to divide. What does it have to do before it divides? In here, we have to address a question. What happens during cell division, during mitosis? So, mitosis is making two copies of each cell. So, we have one cell, but we will divide that cell into two copies. So, both of the cells will have the same exact copy of the genetic material, same copies of the chromosomes. Evet Görkem Sıla, paternal babadan, maternal anneden gelen. Alright, during cell division, we will make two new cells and we will send exact copies of every single chromosome. So for that, before starting cell division, first of all, we have to make exact copies of these chromosomes. What are the chromosomes? Chromosomes are DNA molecule. That means we have to replicate DNA. We have to do DNA replication. So now, I don't know if I can change the color. If I can change, I will be very happy. Oh, what's that? No? All right. So I have to copy these chromosomes. I must make the exact copies of each chromosome. Then I'm making DNA replication. When I replicate the DNA of this chromosome, I will have the same copy of this chromosome. So now I'm making the copy, the paternal chromosomes copy, and at the same time I make the copy of this chromosome too. All right, now I replicated chromosome 1s. At the same time, I also replicate chromosome 2s. So now I'm going to make copy of this chromosome. All right. And also I will make copy of this chromosome too. So this is done, this DNA replication is done during interphase. Before mitosis starts. When they are still in chromatin form, okay? When they are still in chromatin form, before they packed, first of all, we have to replicate the DNA and we have to make the exact copies of each chromosome. Only one thing, the two copies of each chromosome is connected to each other from a special region which is called centromere. So this region is called centromere. Okay? So this and this are exact copies. This and this are exact copies. So there are small little differences between these two chromosome 1s, but these two are exact copies. When we look at this two, so those are, this one and this one are exact copies. Same versions of every single gene exist on these two copies of the same chromosome, paternal chromosome. So each of these paternal chromosome copies are called sister chromatids. Okay, sister chromatid. New term. So after DNA replication, you remember at the beginning we had one copy of paternal chromosome and one copy of maternal chromosome, but then we did DNA replication, we replicated their DNAs, and now we have two copies of paternal chromosome connected to each other from their centromeres. They're bound to each other. They are not separated yet. And those are exact copies. They have the same versions of every single gene. Now, we call products of DNA replication sister chromatids. So this is one sister chromatid. This is the other sister chromatid. They are sister chromatids because they are copies of the same molecule. These are the separating ones, my metaphase. That's correct. We will come there. That's correct, Fatih. All right. Now, this one and this one, this one and this one are sister chromatids. So sister chromatids are products of DNA replication. From the same original molecule. Now, this is chromosome 3, you know. This is chromosome 3. And this is chromosome 3 too. This is the paternal copy of chromosome 3, paternal chromosome 3, but this is maternal chromosome 3. Now, since this is paternal and this is maternal, even though they carry the same genes, they may carry the different versions of the same genes. Because of that, we call these two chromosome 3s homologous chromosomes. So all chromosome 3s are homologous. So what does homologous chromosome mean? Homologous chromosome means two chromosomes carrying the same genes at the same positions. Evet, burayı